Right, so I had a Kahoot for the session we had the last time on Monday. So we'll do this to the end. It's very simple, based on just bacteria, viruses, and fungi. So we could always do that after this session. But we're starting off with some diagnostic techniques in medical microbiology. So that's things that we would do in the lab. So medical technologists use various methods to identify and characterize microorganisms. These include microscopy, where we visualize the specimen under the microscope and just look at their morphology. And we could also do culture and sensitivity testing, where we grow the microorganisms and then test the response to different antibiotics, for example, to establish the best treatment. That is something that we do at the lab, for sure. When we have a sample, for example, a stool or a urine sample, um, and we grow it on a plate like this down there. After we grow the sample, we would take it. I have a slide that shows it when we look at antibiotic resistance, but we would put different antibiotics on the plates, and we could see like which ones are resistant. And based on that information, we could recommend to the doctors which antibiotics would work for that patient. Now, there's also nucleic acid amplification tests, detecting the genetic material of pathogens, enabling rapid and accurate diagnoses. That would be done in more advanced laboratories. In the class on Monday, we did an activity that showed gram-negative staining and gram-positive staining. So I guess you could always look at the recording, or when I share the slides, you could look at the steps. But I could always go back to the slides. Right, so this is just when we identify bacteria, the different steps involved in gram staining, in case you've never done it before. Right. So when you look at a urine sample under the microscope, there are different things that you can see. And these are just some examples. You can see uric acid. And based on what you see, you can make different diagnostic conclusions. Sometimes you have to reinforce that with other tests. But in the case you see something like uric acid, you could think that this is an tumor lysis syndrome, calcium phosphates, magnesium, ammonium phosphates, calcium oxalates, cysteine. There are different casts that you could see under the microscope. In the case that you see something like a red blood cell, then you know for sure that something is wrong with that individual, because red blood cells are too large to filter through the kidney. So you should never see a red blood cell in your urine. So if we do a urine sample and we look at under the microscope and we see something like a red blood cell, then we know for sure that that person's kidney has an issue. And then you could look into other tests to confirm that. The other time you could see white blood cells as well. And depends on the amount, that's when you can make different conclusions. But for sure, red blood cells is a red flag. Also, you can see proteins sometimes. Yeah. So this is strict plating. In the case that you've never done a microbiology lab before, that is what it looks like. This is when you would take your inoculation loop, dip it in your sample, and then you would streak it on the plate. Then you rotate it 90 degrees, do it again, another 90 degrees, and then another 90 degrees. There are different ways that people do streak plating, but this one is the most common one. But in the medical laboratory, to save up, because this is an entire agar plate, to save it, what we would do is we would take a blade and we would split it in half. And then we would plate one sample on this end, and we could plate another sample on this end. So when we split it in half with the blade, we ensure that the two don't cross over, so there's no contamination. But yeah, so just in the interest of saving a plate, because it makes no sense to grow an entire bacterial sample on an entire plate when you could just use half of it. Yeah. So that's what we do. But in just for like at school, that's the one I did. But when we went to the medical laboratory, we would just split it in half and do it that way. So this is what I was referring to about when we plate and we put antibiotics on the plates. So this is what so this plate was full of colonies, and when you put the antibiotics, you would see that it would kill off the bacteria, and based on the size of the circles around the antibiotic, you could like make different inferences. So in this case, no bacteria died. So you could infer that that bacteria strain is resistant to whichever antibiotics were. But just based on the size of the circle on this plate, you could tell that this antibiotic was the best at this one and this one were the best at killing off the bacteria. These have some resistance. These have some resistance. But we would use techniques like this, and then we could recommend to the doctor, okay, so if this was penicillin, for example, you would not prescribe that to the patient because them consuming that would do nothing because the bacteria is resistant. And that is very important because the more antibiotics people use, the more bacteria becoming resistant to them. So tests like this is crucial to establishing which antibiotics are still resistant to whatever strain of bacteria is infecting them. But antimicrobial resistance occurs when microorganisms develop the ability to survive exposure to drugs that once treated them effectively. So antimicrobial resistance poses a significant challenge in healthcare, which leads to difficult to treat infections and increased healthcare costs. So responsible antibiotic use and surveillance are crucial in combating AMR. So it's very important that we don't overuse antibiotics because the more you expose, it's like natural selection in nature. The more you expose bacteria to antibiotics, the higher the probability of one bacteria cell developing resistance to that. And when that bacteria um, establishes that resistance, it's very easy for it to spread because the bacteria could undergo fission, then split in half, and then you get 2, then 4, 8, 16. And just like that, they will multiply, and all these 16 would share the same genome, and they would share that resistance. And if it spreads, then that entire strain, that entire colony would be resistant to that specific antibiotics. So that's why it's very important in the way that you take antibiotics. If you just continue, if you overuse it, like you're basically putting a selective pressure on the bacteria to gain resistance. And once it gains resistance, it spreads like wildfire. So yeah, but that's how it looks like. These are the petri dishes when we put in the antibiotics, and we would see which ones killed off the bacteria. So now we go into antivirals, and these are drugs designed to inhibit viral replication and reduce the severity of viral infections. These drugs specifically inhibit one or more steps of the virus replication without causing unacceptable side effects. So the main target of antiviral drugs, specific events in viral replication identified as targets for antiviral agents like viral adsorption, penetration, and coating, viral nucleic acid synthesis, as well as viral protein synthesis. Specificity for infected cells may occur when virus-specified enzymes activate drugs. So these are some examples of enzymes. So thymidine kinase, so that is used in the herpes antiviral, so yeah, induced by herpes simplex virus. And that's the drug that it would activate, acyclovir. So I'll go back to this slide. It's a very cool slide showing you what, yeah. So these are the steps in the replication of viruses. I don't believe any of you were in the class on Monday, so you would not have seen this slide. Very complicated slide. I just showed it because it looks very interesting. But this is adsorption. So this is when the virus's anti-receptor binds the receptor of the cell and it gains entry. Yeah, it is a lot there. And imagine having to learn this because I did molecular virology last semester, well, in my last year at university, and we had to learn all these steps. It's 7 steps. Um, yeah, but it's just an interesting slide. But it shows, that um, so if you're designing an antiviral, you would aim to target one of these steps. So you could aim at targeting the virus binding onto its receptor. So if you could find a way that you could find an antiviral that would block off them recognizing this receptor, then that's one way of doing it. You could also find a way of, so this is microtubules in the body. And what would the virus would do is it would hijack these systems, so kinesin and dynein, to travel to and from the nucleus, right? So if you could affect this, that's another way of designing an antiviral, or maybe preventing it from getting entry into the cell, into the nucleus, sorry. Or in the case of some influenza viruses, um, targeting the release of the virus. So yeah, that's how, these are the different steps that antivirals would usually aim for. So it's really you understanding how the virus works, and then you could come up with a good way of, countering it. So if it would be like, let's see, so you actually got to work with, no, I wish. Now we only did theory. Um, it's difficult to work with viruses at a undergraduate level, especially in the Caribbean. Yeah, we didn't work with viruses. We just learned the theory behind them. I wish, but nah. Maybe more if you do your master's in virology, then for sure you would probably work with viruses because it's more hands-on. Yeah. But let's move forward to where we continued. We stopped. Yeah. So the limitations with antivirals is that like there's a small amount of antivirus. Ineffectiveness against latent viruses. So for example, herpes is one. Development of drug-resistant mutants and toxic side effects. So the thing with viruses, bacteria, a lot of these simple molecules, simple pathogens is that they replicate quickly and it's easy for them to get mutations. Um, so the more you use antivirals, like I was saying, the antibiotics, it's very easy for you to get like one virus that is if basically resistant to it, and that just renders your entire um, antiviral useless. The other thing is, I think I have here some more reasons why we have a small number of antivirals to date. Specificity, the antiviral drug should only kill the virus infected cells and not host cells or healthy cells. So we have to ensure that whatever process that we're impeding, it only affects viruses. Now that is very important because the thing with viruses is that they use their parasites. So for them to replicate, they use our system. So they use our DNA polymerase. They use like I was showing you a while ago, the microtubules. They use systems that are found within our cells to replicate. So if we create an antiviral that affects those steps, we were essentially affecting our cells. So when you're creating an antiviral, it's very important that you create one that would only stop the virus and not stop the functions of the cell. So that takes time to develop. Many standard pharmaceuticals can be effective if viral enzyme activity is only partially disabled. However, if you partially inhibit, that is not acceptable because it allows for resistant mutants to rise. So essentially what this is saying is that we could come up with pharmaceuticals that would be effective against the virus, but partially. But we can't just go with partially. It has to be fully effective. because like you get more mutants being developed. The third one is that drug discovery is expensive, and I have a subsequent slide that shows like the different steps and just shows how expensive it is. So yes, you could think, okay, we could do that, but it needs to be funded. Some medically important viruses cannot be propagated and have no animal model or are too dangerous. So what this means is that so a lot of viruses like smallpox, hepatitis B, Ebola, you need, like, before you could go to clinical trials with humans, you sometimes have to use animal models, because like you would start with maybe like a cell line, then you move to an animal model, and then when you get positive results, then you'd go to a clinical trial with humans. But if the virus is so dangerous that putting it inside an animal would kill it, then again, that would raise, on the ethical side of things, we would have issues with that. And that's the disappointing thing. So that's why some viruses, like Ebola, don't have any vaccines per se. And this is the slide I was referring to about just the different steps involved in getting an antiviral. So compound synthesized, purified. Um, the next step would be antiviral effect in cells. If it's that point, rejected. Acceptably low toxic in cells. Like you could see the different ones, like antiviral effect in animals, and then acceptably low toxicity in animals, low toxicity in humans. So it's a long process, a lot of different stages that you have to go through before you could actually get a compound that is approved for general usage. Anything that is higher than that would be rejected. And this, it says there, could be expensive. So $100 to $200 million. And also time taken, because imagine having to run this first on cells, then run it on animals, then making sure it's low in animals, run it in humans. Yeah, so that takes about 10 to 15 years. So creating antivirals is a very long process and time taken. That's why when we release those products and people say that it's not safe, they're kind of wrong. Yes, there will be some side effects, but a lot of testing is done before something is approved and brought onto the market for human usage. And this slide just shows the different steps. So it's similar to what I showed a while ago with the different steps in viral replication. But these are the different points that some antivirals can be used. So one particular one is the release of influenza. Because so I think I have a set on influenza, so we'll go back to that slide. Yeah. So influenza has these two receptors on the surface, hemagglutinin and neuraminidase. And for it to enter, the hemagglutinin has to bind onto the sialic acid, that's a receptor. And when it leaves, you'd find that, so when it buds out, so when it leaves, let's go ahead. So in this case, when the virus leaves, it like breaks open the cell. But there are some cases where virus would leave through budding out. So so you'd have exocytosis. So it would bud out of the cell. But when it buds out, the hemagglutinin would still be attached to the sialic acid. So what happens is the neuraminidase, this receptor, would cleave that. So it would remove the sialic acid and allow the virus to leave. So if when we're designing our antiviral, we could prevent the receptor from cleaving that linkage with the sialic acid, then you're essentially preventing the virus from releasing, and that's how you would form your antiviral. So if the virus cannot leave the cell, then it cannot infect more cells, and you essentially preventing it from moving around. And yeah, there are different steps that we're trying to work on antivirals for. So polyprotein processing, so for hepatitis C, HIV, mRNA synthesis in influenza. So yeah, there are different steps, but like I said earlier, it's important that those steps only affect the virus and not the human. So in case of the influenza one, if we're impeding the virus from leaving, that does not affect the host cell. It only affects the virus because it's the one that is attached to the sialic acid. So now for vaccination and immunization, um, we spoke about vaccines yesterday, but vaccines are essential for preventing infectious disease and promoting herd immunity. They stimulate the immune system to produce antibodies without causing disease itself. Vaccination campaigns have successfully eradicated or controlled diseases like smallpox, polio, and SARS-CoV-2 recently. And was just a timeline to show the different vaccines that were created over the years. So in 1796, we first had the live attenuated vaccine, and we had inactivated vaccines, recombinant vaccines, viral vector vaccines, and now we have the mRNA vaccines that was used for COVID, for example. And this is important for establishing herd immunity. A lot of people did not understand what herd immunity is, but essentially if majority of the population is vaccinated against a virus, then the likelihood of someone who never got the virus would be low because people around that individual would be resistant. So the probability of someone who never contracted COVID would be less likely to receive it now because people around that individual is vaccinated against it. And that's essentially how herd immunity works. If we did not get the vaccine and people did not get vaccinated, the likelihood would be higher because you would have no antibodies in your body right now to fight against the COVID-19. So, like I said, COVID-19 came with a lot of controversy. A lot of people were questioning the technology, wondering like, I didn't get vaccinated, and I never got the virus, so this was just a scam. But they don't understand that if everyone in your house except for you got vaccinated, then the likelihood of you getting it is way less compared to if everyone in your household um, did not get vaccinated. So that's essentially how herd immunity works. But now we go into emerging infectious diseases and talking about host jumping. And this was also important in the case of COVID. And this is the one place where the controversy occurred that I could somewhat relate to, although it's not confirmed as yet. We'll get into it. But emerging infectious diseases are newly identified diseases or diseases that have recently increased in incidence. So factors contributing to emerging infectious diseases include human behavior, ecological changes, and globalization. Examples include Ebola, Zika, COVID-19, and globalization is a key one because right now it is so much easier to travel from one country to another. Um, that is makes it a lot um, like easier for diseases to spread. So for example, COVID started in, I believe, China. And because so many people travel from China to all over the globe, you saw that it was able to spread rapidly compared to the 17th century or the 18th century. If COVID started in China, it would be very difficult for a region like the Caribbean to contract the virus because of how long it would take for the person to get there. I'm seeing that travel was mainly through the sea and not in the sky. But right now with globalization, it's so easy for viruses to just spread. So if I have a new strain of let's say COVID and I go onto a plane, I travel tomorrow and I'm going to the US and I sneeze in public, no one has a mask. I could right away spread it there, and then that person goes on to Europe, that person then goes to Africa. So that just shows how easy it is right now for that to happen. Another thing is ecological changes. With climate change, you find that a lot of animals are now spreading where they live, and now they're able to interact with humans more than before. And also with us just starting to cut down forests and getting more space for us to exist in, find out we're now interacting with more animals, and that raises the potential of viruses to spread. But this graphic just shows hop, skip, and jump of the coronavirus. So you could tell that it started with bats. Well, this one is SARS-CoV. That was 2002, 2004. It started with a bat, but then it started all the way back here. So that just shows we had one initial coronavirus, and then there are different branches of it. But the one that was in 2002 started with a bat. It moved to, I think it was a pangolin, I believe this animal is. Can't remember. But then someone interacted with that pangolin, and then was he contracted the virus, and then it was spread in population. Now, the one place I could come up with the controversy for COVID is the intermediate host. We don't know what the intermediate host was for the pandemic. We're not aware. Some people say that it come from a lab. There is no evidence of that. Um, but yeah, that's where I still stick with the controversy of like where exactly that intermediate host came from. But it came from a bat, then got to an intermediate host, and then was contracted by someone, and then that person spread it out through the different populations. And you could tell MERS came from a camel and then went to an individual. Now, for a virus to to the host jump, it has to accumulate mutations that allows it to now affect the host. So that means that the mutations must allow for its anti-receptor to now recognize a receptor that is on the cell of the new host. So when it mutated from this civet, it had to accumulate um, a mutation that allowed it to recognize a receptor in us. So I know for sure for SARS-CoV-2, the receptor is the ACE2 ACE receptor. So the mutation would have allowed that interaction, and then it could spread throughout the population. So this is another example of host jumping and the steps in host jumping. So the first step is exposure of the new host species to the pathogen. So given the different reasons, so like human behavior, ecological changes, globalization, that would allow for the first step, which is exposure to the new host. So maybe now you all of a sudden we're now interacting with ducks. And the interesting thing about that is you find out a lot of diseases, especially the flu, would have come from like some linkage to cattle, well, domesticated poultry animals, I should say. So there's the chicken flu. There is also the swine flu that we had in what year was that? It was like around 2010, around that time. And the reason for that is because we interact with these animals more than we interact with something like a peacock or a porcupine. You don't ever hear peacock flu, or just a random example, because we don't really interact with these animals, but we interact with chickens, we interact with birds, we interact with ducks, we interact with pigs, horses, dogs. So that is why once if those animals were to contract like a variant, it's very easy for us to now contract that because of our close relationship with them. The second step is for the pathogen to be able to infect the new host. So now that we're in proximity with that animal, that host, it must be able to infect us. And that means that we're compatible. So the virus has now accumulated the right mutation to be compatible with us. And then the third step in a successful species jump is the pathogen being able to be sufficiently transmissible between individuals within the new host population. So after we've become compatible, it gets into our system, but it needs to be able to spread. So that receptor that it has must be shared between the entire population. So for example, with COVID, the receptor that they use is the ACE2 receptor. So let me go back to here. So this is the ACE2 receptor, and this receptor is found on mostly all epithelial cells throughout the body. So it was very easy for that virus to spread throughout the population because everyone has those receptors. It's shared. But if it was compatible to, let's say, for example, hypothetically, it was compatible to the first human, but that human had a protein, like the receptor was unique to that individual alone. Then because it's unique to only that individual, even though that virus was spread to other individuals in the population, because they did not have that receptor, since it was unique to the initial person, it would not spread and it would not become a pandemic. But because it recognized a receptor that is common in the population, it could easily spread once it gets exposure to, once the other individuals got exposure to it. Yeah, and that's essentially how host jumping works and what happened in the case of COVID-19. And that concludes my presentation in medical microbiology. Very interesting, especially with viruses, but it's always something that you could read further upon. There is more to it. I didn't want to put too much information to have students overwhelmed, but that essentially concludes it. So if you have any questions, let me know. So I'm not sure if Crystal has any questions. Who else is there? Oh, Shadika. Shadika. Sorry if I pronounced that wrong. If you have any questions, please let me know. Okay, cool. Well, that concludes the presentations for today. Yeah, it was a lot. It was a lot. But like I said, I will share the slides and you could read it at your own time and do some more research. Um, a lot of my topics were pretty heavy. It was really tough to um, fit everything in because I know it's a lot. But I'll share the slides and you could read it at your own time. Yeah, but that concludes the sessions for today, I believe. So we will end here. And if you guys have any further questions, feel free to reach out to me. I'm in the Google Classroom. And I hope you enjoy the rest of your day.